So this is the base space correlation engine, and this is where I had them start. This is my favorite part of, I probably shouldn't be biasing to myself, I'm sorry, Mats, but this is my favorite part of the base space suite of tools. This is where you get to play. This is where we now have access to over 20,000 different studies that we can look at, right? And you don't even need your own data. I can actually, and I'm going to show you, is what we can do is we can put our own data in here and compare it to everything here. But right now with these students, we didn't have our own lab, we didn't have lab space, and I sure as heck wouldn't let, my high school kids around, lung cancer patients. So we had to generate our own data. Here's what we did, and I'm going to take you through that. So under a query term, we can search this database now, and I can put in breast cancer. I said lung cancer, breast cancer, yep. As I'm typing that, they already have these pre-selected categories come up, right? That is the nice thing about this curated data set, is what they have done is they've put text associated with all these different studies. And that if I search breast cancer, it'll basically find me all those studies. If I hit return, oh, wait a minute. If I hit that button, what it is now going to do is basically pull up all the references that it has for breast cancer. Any study, anything that had breast cancer associated with it as a key term. And right here now, it's just giving us an overview. That if we look here, based on what the phenotype that I put in, it's bringing out some of the genetic markers associated with breast cancer. And we could look at any of these. Just a quick look here, these are all the heavy hitters of cancer. Cyclin D, here's an estrogen receptor, obviously relevant to breast cancer. ERB2, growth factors, cadherin molecules, these are how the cells bind together, and P53, I think a lot of people have heard about P53. Which is good information, and we could play around with some of this stuff, but what I want to do is I want to build my own data set. Right? Because I'm not just interested in just breast cancer as a generic term. I want to know what's different between stage 3 versus stage 1 or stage 2. Okay? So I can go to curated data or curated studies. This is the second one. Can everybody see that? Is it washing out too much? Is it okay? Is it okay? Okay. That doesn't help. Okay. When I do that, when I go to curated studies, it is bringing up 974 studies that have breast cancer somewhere as the phenotype. That's a lot of data. And I'm sure, when I teach that, I teach a session in the summer up at Snowmass to clinical oncologists. And I asked them to type in their cancer, and a lot of them are amazed. At first they go, well, I work on a special cancer, it's not in here. Usually that's their first reaction. No, I'm working on blah, blah, blah. And they'll put in their keywords, and sure enough, like 2 or 3 other studies will pop up, right? We all think we're, working on, all our, everybody's research is unique and no one else does what we do, but guess what they do? If you look, I swear they do. Okay, so we've got breast cancer. Now we have some potential studies that we can look at. I want to filter this a bit, right? Again, I said that I'm really interested in stage 3 breast cancer. So I go to organisms here, and I can say, I only want to see people. I go to data types, and you have, again, all of this information is in here. RNA expression, you've got SNPs, methylation, copy number, mutations, and you also have microRNAs, and you also have protein studies in here. Again, everything on that slide that I told you we can measure is all in here, right? You can pick whatever you want to look at. And in fact, what I like to do is pick several, and then I kind of combine them, but we'll talk about that later. Okay, we can go to keyword here. And I'm going to put in stage 3 because that's what I'm interested in. And I'm going to go to advanced. And I'm interested, I don't want to see stage 3 versus healthy tissue. I want to see stage 3 versus other lesser forms of cancer. So here's what I'm going to do. On my experimental design, I want to say see disease versus disease. And then I can, and then I could say, and then all I want is from the tissues, I want to see it from the primary tissue, fresh or frozen. I can pick blood, so that would be good for a biomarker to see if potentially somebody got breast cancer, a biomarker in the blood. Lots of other things, cell culture, again, primary cultures, bone marrow fraction. I'm going to hit apply filter. Oh, I didn't hit my RNA-seq, sorry. Go to data types, RNA-seq, or RNA expression, I mean. So now we're down to 110 studies. What I can do, and that's what I had, so basically I took the kids through there and I'm like, okay, let's make our own meta-analysis. Let's generate our own G-list. And what I don't want you kids to do is use just one study, because what if that study is horrible, right? I want you to get a bunch of studies, and then we're going to take the average of all of that, and that's going to be our data set. And by the way, the final data set was probably one of the best cancer, gene lists I've ever seen. So here's how you make a meta-analysis. So basically, you can go through here, and you can see what kind of comparisons are in this study, and you can actually pick the ones you want. So here, these are all primary breast tumors. Here is grade 3 versus grade 1. I'm going to select that one. And you look down here, there's stage 3 versus stage 1. Yeah, why not? Keep going. Where am I looking? Grade 2 versus grade 1. Nope. Grade 3 versus grade 1. Yep. As I am doing that, I am basically adding these gene lists to my meta-analysis. Let me get one more. Grade 3 versus grade 1. Yep. As I look here, here's what it's telling you. That I am selecting this list. In this RNA expression study, when they looked at grade 3 versus grade 1 from primary breast cancer tissue, I believe so, they found 5,439 genes that were different. I'm now taking that to my meta-analysis. Okay, we can go back up here. These are now all collected in the meta-analysis. This is on the right. I'm going to click on this. Here are all my 3 gene lists, or 5 gene lists, I'm sorry. It's like a Monty Python 3 and 5. Makes sense. And I can reorder these any way I want, right? I can take these, I can put this one on top, I can get rid of this, remove any of these, I can add them on the fly. That's what makes this nice, is we are now in a curated system. I don't have to download this and analyze this myself. I trust Illumina. Illumina sequences 95% of everything on this planet. I'm going to trust that they did a good job of normalizing and processing this data. So now I get all the fun stuff, right? I get to search this stuff. So we can hit the meta-analysis. Again, this is showing you what gene lists you are comparing. And then I can go to my gene results. And this gives you the genes in all those gene lists. And it'll tell you whether that gene, integral membrane protein 2A, was differentially expressed in all those different studies. If there's a line here, if there's a color, that means it was differentially expressed. And you can go to each one of these different studies and see which one it was, or gene list. If it's green, that means that gene was downregulated in that study. If it was overexpressed, it'll be red. Again, here's the specificity. Here's what I like to do, and this is what I had the students do. And you can see that, for the most part, these agree. right? Even though labs from thousands of miles apart, all over the country, maybe all over the world, looked at the same grade 3 versus grade 1, they're coming up with the same genes. It's kind of like a poll that if all 5 polls say that I2M2A is going to win the election, it's probably going to win the election, right? Same kind of thing. So what I had them do is basically we generated this gene list, and I just did it very quickly, but I had one of my students, he had a gene list of 58 different gene lists that he used for his meta-analysis. It was insane. I was like, you can't use all that, man. You gotta cut it back a little bit. It took him so long to process. But what I had them do, though, is I would make their own gene list, and what you can do now is I can export this as an Excel file. So if I hit that, it should be going. I'm going to export this as an Excel file. So I'll have all those genes. I'll have all their p-values on each different study and all its fold changes. There it is. Ah, cool. So these are the genes that were in those 5 things. And so what I had them do, you can look at all the different p-values and fold changes in each of the different studies, which are listed here. But what I had them do is basically calculate what was the median fold change for all of their studies that they looked at, number 1. Number 2, what was the median fold change? That's how I'm combining all this data. Once I did that, I made a column, and now I can take that list and import it back into Ingenuity. I know I've been kind of going through this, and no one's been asking any questions up until this point. Sometimes when I get talking, I feel like people are just like going into a coma, like too much information. No questions? Yeah.
Why did you use the median and not the mean?
I didn't use the mean because there's that's a very good question, is outliers, right? A lot of my studies, or not only that, but sometimes they might not always agree with each other. So maybe I have four studies where it goes up and then maybe one where it goes way, way down, right? I want the median, so I'm getting most of those 4 there. Everything this is just, I view bioinformatics, my role as a bioinformatician is I'm a guide, right? And it makes it nice because, I don't have to prove anything until you do it. I'm surfing the numbers, right? That, you send me out in advance to kind of scope out the territory, and I say, hey, here's a trail. Let's go this way, right? The nice thing is, this works is if you've come up with a target and you validate it at the lab, right? That's when all this stuff works. And I'm not here to, blow smoke somewhere to tell you that, this is the end all and be all of science. This is to guide you to where to go. And I think it's very good information. Okay, so this is what I had them do. We downloaded it. So we had a list we used 24 different studies looking at grade 3 versus grade 1. And as you can see here, I can determine how many gene lists I can require, how many gene lists it appears in. Here, you go, these genes were in 5 out of 5, 4 out of 4, 3 out of 3, etc. I required that was in at least more than half of the 24 data sets. So it had to be differentially expressed in 13 out of 24. And here is where you can upload your data.